Hello and welcome back to my channel BioStudies. Myself Komal, your biology tutor. In this lecture, I am telling you some basic and important questions from the chapter Anatomy of Flowering Plants. So let's begin. Question number 1 is, what is plant anatomy? Anatomy is the study of internal structures of plants, focusing on tissue organization and function. So it's a very basic definition of what plant anatomy is. Next is, what are bulliform cells and their function? So bulliform cells are basically modified epidermal cells. These are large epidermal cells that is present in the monocote leaves. What are their function? Help curling the leaves under water stress to reduce transpiration. So to reduce the water loss, these type of cells, modified epidermal cells, that is bulliform cells, are formed in the monocote leaves. Next question is, define the vascular cambium. It's a very important question. What is vascular cambium? So it is a lateral meristem that is formed, or you can say that it will form a cambial ring between xylem and phloem. So there is a xylem and phloem. It will form a cambial ring between them, and it's a lateral meristem forming a cambial ring, and that ring is responsible for secondary growth. Next question is, what is core cambium and what does it produce, right? Core cambium, also known as phloem. Phloem is a type of lateral meristem in plants that is responsible for producing cork cells on outside and secondary cortex on inside. देखो, so there are phloem, phloem and phloem. These three terms are again very important. So phloem, phloem and phloem. So core cambium and secondary cortex. Phloem is cork, phloem is core cambium, and phloem is your secondary cortex. So this layer, this meristematic layer is formed between these two, that is core cambium, right? So that is the location. And what is it? It is a type of lateral meristem. Easy? Okay. Next is, which tissue forms the fibers used in jute ropes? जूट रोप्स में जो फाइबर्स बनाता है वो कौन सा टिश्यू होता है? The sclerenchyma cells form tough lignified fibers in the jute ropes. So that tissue is sclerenchyma. Next question is, differentiate anatomy of dicot stem and monocot stem. Dicot stem, vascular bundles are present in ring. There is a presence of pith and cortex. In monocot there are scattered vascular bundles. Secondary growth is not there because there are no cambium is not there. There will be no secondary growth in monocots. There are secondary growth in dicots because cambium is there. Cambium is responsible for secondary growth. There are vascular, there are core cambium. Okay. Next is, compare between these two, like dicot and monocot leaves. Again, dicot dorsiventral, distinct palisade and spongy, means you can differentiate between the two, palisade and spongy mesophils. In monocots, these are isobilateral. Mesophils are not differentiated. Okay, it's a very easy again. If you know the diagrams properly of dicot and monocot leaves, then it will be easy for you to answer that type of questions. Okay. Next question is, what are plasmodesmata? Cytoplasmic strands between adjacent plant cells enabling transport of substances. Okay. What is secondary wall? It is a thick lignified cell wall deposited after cell maturation, adding structural strength, right? So it's a lignified, here lignin content is there, lignin content की वजह से ही strength is added in this particular wall. Okay, so yeah, so that's all for this video. It's just a part 1. Part 2 भी जल्दी आएगा. If you have any doubt in any question, put your doubts in the comment section. Okay, thank you. Thanks for watching.